very much, Kate, for the introduction, and thank you all for this opportunities to share our recent work on the whole genome methylation profiling of central nervous system tumors, and validating this assay for clinical diagnostic with enhanced tumor classification using the DNA methylation signature. Okay, here's my title page, and I don't have any disclosures. Okay, first of all, a little bit of background and why methylation analysis for CNS tumor classification. So DNA methylation play an important and dynamic role in regulating gene expression. It allows cells to acquire and maintain a specialized state, and aberrant DNA methylation and its impact on the gene expression has been implicated in many disease processes, including cancer, neurological disorders, aging, and development. So as you all know, the DNA methylation is an enzyme-mediated epigenetic modification to DNA that add a methyl group to the cytosine base. So in eukaryotes, methylation occurs mostly at CG dinucleotides or CPG sites on both strands of the DNA. So the second point is the DNA methylation is a robust and stable biomarkers of cell origin and disease. DNA methylation is highly related to cell fate specification during cellular differentiation, so which is driven by a gene expression program depending on the transcriptional control. And the DNA methylation at the gene promoter and enhancer regions is crucial for coordinate such programs. And in cancer, the genome-wide DNA methylation pattern, also known as methylome, is a combination of both somatic acquired DNA methylation changes and a characteristic that reflect cell origin. The latter property enable, for example, the tracing of the primary site of highly differentiated metastases of cancer of unknown origin. So the third point is that DNA methylation-based tumor classification has emerged as a promising tool to dissect tumor classes and to improve diagnostic accuracy. So about more than 50% of the diagnostic entries in the current edition of the WHO classification of CNS tumors included DNA methylation profile as an essential or recommended diagnostic features. The CNS tumor has historically been classified using histology with H&E staining sections and IHC expression of lineage-associated proteins. So the biggest challenge to this is significant inter-observer variability. So in some reports, up to 40% discordant diagnosis, especially in certain subtypes, which are more difficult to classify, such as astrocytomas. So in the in the past 2 decades, there has been a lot of work done to uncover the genetics of CNS tumors. And as a result, the recent WHO guideline was revised to include some of the molecular typing and or DNA methylation profiles. And CNS tumors are clinically extremely diverse, ranging from benign neoplasm that can be cured by surgical alone, such as pilocytic astrocytoma, and to highly malignant tumors that respond poorly to any form of treatment, such as glioblastoma. So being able to correctly diagnose the subtype of CNS tumors is crucial and has a huge implication for prognosis and treatment plan. So to translate the methylation array data to practical use, a variety of analytical and computational steps need to be performed. So much work has been done to establish a comprehensive DNA methylation-based CNS tumor reference cohort. and which is pioneered by groups at the German Cancer Research Center in collaboration with scientists and physicians all over the world. So one of the most important contributions from these studies is the implementation of a machine learning-based classifier to prospectively evaluate new samples. So this classifier, also known as Heidelberg classifier. As shown here in panel A, 82 CNS tumor methylation class and the 9 control tissue methylation class were included in the reference cohort with 2,800 samples. So the methylation class are grouped by histology and color-coded and listed here. And here is the t-SNE plot to show the cluster of tumors based on the methylation data. So a frequently used and practical approach to delineate tumor type with methylation data is a dimensionality reduction. Similar to principal component analysis, these unsupervised algorithms reduce high dimensional data. For example, thousands of brain tumor samples, each with thousands of data points, to a lower dimension like 2D or 3D for visualization. So one of the most widely used technique is t-SNE, stand for T-distributed stochastic neighboring embedding. So panel B show an unsupervised clustering of reference cohort, including 2,800 samples use t-SNE dimensionality reduction. Notably, methylation results leads to the diagnostic revision in a significant proportion of cases, which is about 12%. And a worldwide cooperative network has been contiguously identifying and tracking CNS tumor classes. So far, there were more than 30,000 CNS tumor analyzed. So by the way, all the data used here was generated using Illumina methylation microarray, also known as BeadChip. So to implement this classifier in the diagnostic practice, the classifier was validated by an independent prospective cohort of more than 1,100 diagnostic samples. So the pathological diagnosis was established by current pathological standards and compared with classification by methylation profiling. So cases was categorized as confirmed diagnosis in 76% and establishing the new diagnosis in 12% and the misleading profile or no match to define the classes in about less than 1%. So over the years, the classifier has been contiguously modified with more tumor entity being recognized by each update. So, and such evolution, will also continue in future. And as our knowledge on the novel classes grows, so currently, the classifier is used in the clinics in some clinics in U.S., Germany, Netherlands, and Denmark, UK, and Japan. So now let's look at the discrepant cases in this study. So the methylation results leads to the diagnostic revision in 12% of the cases. So this finding was confirmed in 5 external centers with reclassification rate ranged from 6% to 25%. And the discrepancy between the pathological diagnosis is on the left, and the methylation profiling in the middle. And for 129 cases, the histological and molecular assessment resulting in a change in the initial diagnosis with the formulation of a new integrated diagnosis for 92 cases listed on the right. And so for those 92 out of 129 cases, This involved a change in the WHO grading, with both downgrading shown as the blue lines and upgrading shown as the red lines. So why choose Illumina methylation EPIC array for DNA methylation profiling? So sometimes they call it the methylation array, or sometimes they call it EPIC array. It's the same thing. So there are a variety of methods that exist to quantify DNA methylation, each with various application in clinical oncology. For example, restriction endonuclease digestion, affinity enriched methods, and whole genome bisulfite sequencing. Although whole genome bisulfite sequencing can quantify the methylation state of the about 29 million CpGs in the human genome, it is currently extraordinarily expensive and computationally intensive. So a comprehensive and cost-effective alternative is the complementary use of bisulfite converted DNA and the microarray technology. So I listed three main reasons here. So Illumina Infinium HD technology provide comprehensive genome-wide coverage. And this robust methylation profiling platform can provide high quality data with internal sample dependent and sample independent quality controls with high assay reproducibility. I can go over these in more detail next. Also, it has a streamlined workflow and a compatibility with FFP samples with demonstrating robustness to pre-analytical factors. And integrated analysis software and classifier that I mentioned earlier, which used Illumina methylation array data. The second point is the TCGA data. Of the about 11,000 cancer sample cataloged in the Cancer Genome Atlas, majority sample had the methylation profile, has methylation profiled by Illumina array technology. Only 39 has been profiled with whole genome bisulfite sequencing. So we have abundance of reference methylation data. And regarding the coverage of the Illumina methylation EPIC array, also called the BeadChip, it offers a unique combination of expert selected comprehensive genome-wide coverage, which contain 850K methylation site across the genome. So these contain probes pre-selected for biological meaningful regions of the genome, including CpG islands and adjacent regions as shown in the figure here. And it also includes non-CpG methylated site identified in human stem cells. And the differentiated methylated sites identified in the tumor versus normal across multiple forms of cancer and the tissue types. And the encode open chromatin and enhancer regions. And also the array also targets the enhancer identified by the Phantom 5 project across the tissue type, and the DNA hypersensitive sites and the microRNA promoter regions. So this coverage can give a pan-enhancer and the coding region view of the methylome that can be used for epigenome-wide profiling. Okay. So regarding the, let's see now, let's talk a little bit about the probe design and how this technology works. So there are two types of probe design and assay designs, or assay design, and Infinium 1 and Infinium 2. So for the Infinium 1 assay design, it uses two type of bead, use two bead types per CpG locus, one each for the methylated and the unmethylated state, shown here. So the 3' terminus of each probe is designed to match either to the protected cytosine by methylation or to the thymine base resulting from bisulfite conversion and whole genome amplification. This is unmethylated design. So the for Infinium 2 design use only one bead type with the methylated state determined at the single base pair extension step after hybridization. So the 3' terminus of the probe complements to the base directly upstream the query site, a single base extension resulting addition of a labeled G or A, complementary to either methylated or unmethylated state. So as you know, the bisulfite conversion reaction is a chemical reaction which converts all unmethylated cytosine in the DNA sequence to uracil, and which eventually converted to thymidine post amplification in the assay. So the methyl group at the methylated cytosine prevented the deamination, blocked the bisulfite conversion. So after bisulfite conversion, the unmethylated C becoming T, and it will match with unmethylated probe, have a 3' end extension for Infinium 1 probe. And for Infinium 2 probe, the one single base pair extension will add an A at that position. So for methylated C, it will match with the methylated locus in Infinium 1 probe, have a one base pair extension after that, and or the added G for the Infinium 2 probe. So the basic assay output for a given CPG are intensity for methylated signal and unmethylated signal. So the beta is the proportion of methylated signal to total signal with a small constant added to the denominator for stabilization. So for methylated CPG, IM is high and IU is low and near 0. Therefore, the beta value is close to 1. And so show here. And for the unmethylated CPGs, IU is high and IM is near 0. And then the beta value is close to 0. So this is the distribution of probes, for a sample, and you usually have two peaks, one near 0 and one near 1. So this is the general process of methylation epic array analysis, which is very similar to the cytosNP microarray analysis we use for copy number evaluation, except that there is an additional bisulfite conversion step. And of course, the data analysis is very different. So this chart from the Pratt paper gave a very nice overview of the workflow and the current diagnostic application of DNA methylation profiling and its role in the surgical neuropathology. And so basically, in the Illumina epic array are compatible with both fresh frozen tissue and FIPE material. So following the extraction, the DNA is bisulfite converted and hybridized to the BeadChip overnight. And DNA from FIPE tissue needs to be treated with FIPE restore kit prior to the hybridization. And then the following day, there are several steps, including single base pair extension, target removal, and the staining of signal amplification, staining for the signal amplification. Then there are 3 rounds of washing steps. So after the slide is dry, the fluorescence signal are then processed with eye scan readers, and which generate 2 separate data files, one for each color channel, red and green. And then the data analysis is done using the 3-program package. The Genome Studio can convert the raw image file to probe density, and generate a control dashboard for QC metrics. And the signal processing methylation-based tumor classifier and the MGMT promoter methylation analysis were carried out for clustering and data visualization using the in-house tumor classifier software pipeline developed by the DKFZ and the German group. We will be able to communicate with them and implemented this package in-house. And so for the calculation of the CNV, the methylated and unmethylated signal intensity are added together, and a ratio is formed against the healthy reference samples that has a flat, a normal genome. So this copy number ratio is then plotted in a graph according to the chromosome position, location. This is a R-based package, and it's used for a copy number analysis. So the classifier provides three reports, three elements. One is methylation-based classification, and also chromosome copy number plot, as well as the promoter method, the MGMT promoter methylation status. So finally, as with any diagnostic assay in the surgical pathology, methylation profiling should be interpreted in conjunction with all available clinical data, including history, imaging, histology, and other supplementary molecular testing. So here's our samples. First, let's look at the parameters for QC applied in our pipeline, the QC summary. So there are parameters for overall performance of the analysis. There are also parameters for each of the steps in the process, and they're listed here. And we have tested 116 samples, including 106 tumor specimen and 10 normal control samples. So this figure here summarizes the QC data with, x-axis for the QC parameters and the y-axis for the sample tested. The value near 1 show as the blue for pass QC and the yellow for non-pass QC. And you can see that majority of our tested cases passed QC. And if you look at the beta curve, and these samples also show the typical pattern of beta value distribution with peak near 0 and one peak near 1. So total, the sample we have passed QC is 109 tumor samples and 7 failed pass QCs. So for the sample that passed QC, we had 99 tumor cases, including 50 adult cases and 49 pediatric cases. And this includes 14 histology categories and 41 tumor entities listed here. So we have some, we have more samples for some categories than others. So we also had 10 control cases, which include 6 CNS tissues and also 4 non-CNS tissues. So the results. So the prediction based on the methylation profile correlated with the histology and the molecular diagnosis in the validation cohort. Dr. PJ Semino and helped with the correlation analysis for adult cases, and Dr. Bonnie Cole and Vera Paulson helped me with the correlation analysis for the pediatric cases. So the pathological diagnosis, was established by current pathological standards, and which we compared with classification by methylation profiling. So first, all 10 control cases was classified as control, a normal tissue, and the 96 out of 99 tumor sample had the methylation classification that are concordant with the histology and the molecular diagnosis, such as oncoplex and FISH analysis. And the 3, so this is given this number, so our which result analytical specificity is 97, and analytical sensitivity is 100%. So for the 3 of cases that are discordant, I list them here. And 2 had high sub-classification scores based on the methylation. And the pathologists agree that the methylation provided a more accurate diagnosis for these 2 cases. So for this case showed in green box, had a relatively low classification score by methylation, so which gave a misleading, which is have no match actually to a defined class. So this case needs a further examination. This is another way to show that our validation cohort. So this is the t-SNE clustering of the analyzed cases against the DKFZ reference data set. The reference cases are colored according to the respective methylation classes, and our validation cases are shown as dot with the black circles. And you can see that our validation cases are clustered well with CNS tumor methylation classes of the reference cohort. They are very distinctive clusters. Okay, now how about the reproducibility? So to examine the reproducibility to determine the precision and the intra-precision of the methods, 14 cases was also processed and analyzed twice in different batches. And 3 cases was processed and analyzed twice in the same batch. They all had concordant results with high classification score. So one way to visualize that is to see the heat map of top 1,000 variable CPG sites. So the y-axis is the beta value of the probe, and with the dark blue show as unmethylated and the yellow as methylated. And the x-axis is a different case, different type of cases, and 1, 2 show the duplicates for the same sample. And you can see they are highly correlated. And then we also did that for intra-batch duplication. They correlated even, they even more correlated than the inter-batch, which is expected. So how about DNA requirement for the assay? So to determine the minimum quantity of the DNA required for methylation-based classification, we perform a methylation profiling using the various quantity of tumor DNA input, ranging from 150 nanogram down to 25 nanogram for case, and this for case 1, and case 2 is 75 nanogram to 25 nanogram. So all of 7 all the 7 tests, 6 passed QC, and the test with 25 nanogram in the case 1 did not pass the QC. Show here, this is the QC matrix. And so the copy plot also show that the copy number alteration are detected for 150, 75, and 50, but not for the 25 nanogram input DNA for case 1. For case 2, copy number alteration detected for all 3. So taken together, the minimum DNA quantity requirement is above above 50 nanogram. This one just to show the this just show the case which with correlation between the DNA input and the classification score. So you can see that this is a failed there. And also we showed the mean of probe deviation, which is increased with less DNA. Tumor content requirement. So we plotted the cases with clear tumor content estimated based on our cytosnip microarray analysis, or variant allele frequency based on the mutation analysis with estimation by pathologists. So the lowest, so here is the score past 0.9 threshold, and the lowest tumor content is 30%. So this is our validation cohort. We also did the dilution test. So to determine the tumor content required for methylation based on the classification, we performed methylation array profiling for 3 cases, composed of varying ratio of DNA extracted from the tumor and a non-neoplastic sample. And the classification score above 0.9 was achieved for malignant melanoma DNA, which show as blue, and down to 10% of tumor DNA, and medulloblastoma DNA down to 20%. Here, so that is below the 0.9, and but for the meningioma and with DNA down to 40% DNA, show as a gray dot. So although those classification score are below 0.9 threshold for low tumor content, they are still classified with their respective methylation class. So taken together, the minimum tumor content requirement is 30 to 40%, and the optimal tumor percent should be above 70%. We also did copy number evaluation and validation by methylation array. And so we have 22 cases, which has been characterized by both cytosnip 850K microarray and EPIC array. So the cytosnip 850K is the array we use for copy number evaluation, which is a gold standard. So there are total about 328 copy number changes by cytosnip array, and including 183 copy loss and 145 copy gain. And for the EPIC array, 291 copy number alteration was detected, including 87, 159 copy loss and 132 copy gains. So this is the plot show the size of the copy number changes and the percent tumor content on the y-axis, and the blue dot. Actually all 291 copy number alteration by EPIC array was detected by the cytosnip microarray, but there are some copy numbers by cytosnip microarray was not detected by EPIC array. So this is the plot showing that the blue dot is detected by both platforms, and the red one is only detected by cytosnip array. And you can see that the red dot is the distribution of the red dot is usually below 25% of tumor content and the small copy number changes. So if we make a cutoff at 200 kb, for example, and tumor content at 20, and for above 250, about 200 kb and 20% tumor content, and EPIC array can detect 96% of the copy number changes for loss and 94% for gain. But when the tumor content is low and this drops, the loss is can only detect 25% the EPIC array and 43% for gains. So now let's go to the MGMT methylation. And so the methylation status of MGMT gene is important predictive biomarker for benefit of alkylating agent therapy in glioblastoma. So study using the same array platform and probe coverage for MGMT gene interrogate 176 CpG annotated for MGMT gene with 18 probe. You can see here the numbered probe from 1 to 18, and this here is the genome coordinates, and the green little bars show the CpG island and also the CpG sites, and different horizontal bars show the different assay, which region they interrogated. And so in the panel A here, show the correlation between MGMT gene expression and M value of 18 CpG methylation probe from the GBM cohort they used. So the methylation status probe 10 and 16 below this gray line are negatively, the methylation status of those two probes are negatively associated with gene expression. So panel B show the significant association between overall survival and the CpG methylation of distinct probes, which display with the p-value above the dotted line. So the dotted green line is corresponding to the threshold of 0.05, and so includes 8, 9, 10, 11, 12, and 16, right? So this one is panel C show the association between MGMT promoter methylation classification based on the other methods. And so again, the Chrome 10 and 16 show highest correlation. So overall, the probe 10 and 16 are most correlated with MGMT expression and survival. So their approach using a logistic regression model called MGMT ST27 with probe 10 and 16 was deployed. So this classification, they had a formula to calculate the scores, estimated scores, and establish cutoffs. So the classifier adopted this approach because same probe was used by this paper and also the array platform we are using now, and to estimate the status for MGMT promoter methylation. So this is the formula for MGMT promoter methylation prediction scores. Okay, so with the help of Abby and Eric, so we were able to compare the predicted MGMT score by EPIC array with their RT-PCR assay results for 14 cases. So the panel A here show the beta value of 18 probes in the MGMT region by methylation array analysis. The light blue is for methylated, so this is the beta value. The light blue is for methylated loci with higher beta value, and the dark blue is for unmethylated loci with lower beta value. And you can see that the methylation status across the CpG side are not uniform. So this is the x-axis is the cases, and then this is the probe, and then their beta value. So the CpG methylation probe in the blue box show here. So the blue box are the low, let's see. In the blue box, the not significantly correlated with MGMT expression or overall survival, but the previous study I showed just earlier. And the CpG methylation probe 10 and 16, which is the most correlated with MGMT expression and the overall survival, is shown in the red. So in the red box here. And the CpG methylation probe 10.6, which move, and their corresponding beta value also plotted underneath in the panel B here. And these values are used to calculate MGMT promoter methylation prediction scores. So the panel C here show the correlation results between the MGMT prediction score by array and methylation status by RT-PCR. So the cutoff for RT-PCR assay validation by Eric and Abby has a delta CT higher than, so this here is the cutoff value, higher than 7.5 as negative here. So this is cutoff for negative. And then between 5.5 and 7.5 as indeterminate, and the positive for lower than 5.5. So you can see here, those four are negative by RT-PCR, indeterminate, and positive by PCR. So the cutoff for by EPIC array is 3.6. So for methylated site, the value is larger than 3.6, and with the CI lower boundary should be larger than 3.6. So this is the mean value, and here is the CI low and CI high. So we call it indeterminate when the the mean value is larger than 3.6, but the CI lower is less than 3.6, and unmethylated when it's lower than 3.6. So you can see that here's concordant cases and here are concordant cases, and the indeterminate cases by RT-PCR showed to be positive by methylation array. We only have one indeterminate cases in this 14 cases we tested, which have really big CI, lower and CI high boundaries. And the discordant, MGMT, discordant MGMT status, can be compromised by many different variables. For example, methylation is because methylation is a contiguous variable, and the most threshold in the lab are set arbitrarily, and also methylation profiling can vary across the sample regions within the single tumor, as shown here with a patchy pattern of a beta value. And also, variable detection can occur across the platform. And, different platform was used. For example, here, methylation specific PCR versus methylation array, and it could be variable band methods. And so overlapping but different location of CpGs are interrogated by different assays, also another factors. So, all those kind of affecting the determination of the methylation versus unmethylation of MGMT promoters. Okay. All right. Am I okay with time? Now let's look at the data for some specific cases. So medulloblastoma methylation subgroups shown here. So this is a TSME clustering of analyzed medulloblastoma cases against the DKFZ reference database. The reference cases are colored according to the respective methylation class. Our validated cases show as dot with black circles, and all medulloblastoma validation cases cluster well with medulloblastoma methylation subclass of the reference cohort with a very high classification score. And here's the heat map of top 1,000 variable CpG sites across 4 subgroups, which show the distinctive methylation patterns for each subgroup. So, the y-axis is the cases and the beta values. And for example, as the HH subgroup, for example, have a distinctive methylated and unmethylated, the overlapping partly with group 4. And group 3 and group 4 have share methylation pattern for some loci here, but differs in other sites. And the molecular subgroup in medulloblastoma exhibit a distinctive transcriptional epigenetic signature that define clinically relevant patient subset. So a combination of molecular analysis, such as DNA methylation and the mutation analysis, and the morphological interpretation can provide optimal prognostic and predictive information. So, and some, these genetic alterations are unique to each group, and others, are shared and overlapping between subtypes like isodicentric chromosomes 17. And so here, just the copy plot show different subtypes and their signature as minus, monosomy 6 in Wnt, and 9p deletion, containing PTCH1 genes in the SHH, and isochrome 17 here. And this is just the zooming in of isochromosome 17 to show the breakpoints. And also, gene amplifications and some distinctive deletions in group 3. So medulloblastoma, are represented by a higher number of reference cases and are among the best defined group in the classifier. And so it believed that a failed classification should be a prompt consideration of other tumor classes not included in the classifier, such as defined, such as non-defined embryonal tumors or metastases. Another example is IDHglioma. Again, the T-SNE cluster show our validation cases, and here is the copy number plot by methylation array analysis for our oligodendron glioma case with 1p19q whole arm co-deletion. While this deletion is usually evaluated by FISH analysis, we find evaluation of the copy number pattern by array is much more reliable. And also the heat map for 1,000 variable CPG sites across the OIDH, oligodendron, astrocytoma, IDH. This is a IDH mutant and IDH wild type. And you can see that the classifier recognized tumor with a IDH mutation associated CPG island and methylator phenotype very well, and therefore both IDH1 and IDH2 mutated tumors are detected independently of the mutation type, including variants not detected by, for example, the IDH1 R132H antibody. So the association of the DNA methylation class IDHglioma and the presence of IDH mutation in the glioma is so strong that the methylation class can be interpreted as a proof of IDH mutation status. And the last example I'm going to show here. Hope I have. Oh, wow. Okay, maybe I should skip this one. Let's just skip this one. I think time is short. Just go to the summary of the utility. So to summarize the utility of genome-wide methylation profiling in CNS tumor classification, as I pointed out earlier, it can help reduce tumor misclassification and the interlab variability, improving precision overall. So the Heidelberg experience suggests that for the pediatric CNS tumors, there are several reasons for examining all pediatric CNS tumors by methylation analysis. For medulloblastoma and ependymomas, therapy decision in Heidelberg are often more heavily influenced by methylation class rather than WHO diagnosis. And for the tumors from adult patients in need additional subclassifications, such as tumors with the morphological aspect of astrocytoma, but but lacking IDH mutation. So in most instances, these patients turn out to have glioblastoma glioblastoma based on the methylation class and the C and D patterns. So another typical tumor to be included in the morphological astrocytoma with IDH mutation and maintain the nuclear ATRX expression. So this latter group frequently turn out to be oligodendroglioma with evident 1p92 co-deletion. And they regularly also submit low-grade gliomas and glioneuronal tumors to methylation analysis. They find this was very helpful in distinguishing between ganglioglioma and the pilocytic astrocytoma and the PXA. So for histologically ambiguous and complicated cases, even when a match with high confidence score was not reached to fit in any recognized group, the main utility here is to ensure that it does not fall into any specific and or recognized group that is somehow missed. So finally, it can help detect novel tumor entities and enable more or better clinical trial. So here's just the summary, just reiterate what I just described. We have validated this platform and the classifier for diverse CNS tumors, including 14 histology category and 41 tumor entity with high analytical specificity and sensitivities. And the data can be used to help CNS tumor classification and risk stratify patients. And so here is what we report. Usually is included the classification by methylation and MGMT methylation status, and also the copy numbers we found by this platform. So what's the future projects? The future projects involves applying DNA methylation profiling and classifier to different cancer types. For example, recently there's a sarcoma classifier just becoming available. And like a CNS classifier, you use machine learning classifier algorithm based on the array generated DNA methylation data, and it is trained using a data set of more than 1,000 methylation profiles from pre-characterized cases. This consists of 62 tumor methylation class, a broad range of soft tissue and bone sarcoma subtypes. So it demonstrated that the potential of the DNA methylation-based sarcoma classification for research and the future diagnostic applications. Finally, this is truly a teamwork, and I have many people to thank for. First of all, Dr. He Fang, and she's not only a good geneticist, she's also very well versed with our language, and so she's instrumental for us to implement this classifier in-house. So, and also Yu Wu and Yuhua Liu, who has performed all the bench work and participated data analysis. And I also like to thank PJ and Bonnie, who selected the validation cohort for us and did the correlation study after the methylation profiling. And Vera also did a lot of correlation study for us, and Vera and Tina provided a lot of OncoPlex data for us to do the correlation. And also Eric and Abby, who provided the RT-PCR MGMT results for us to be able to correlate for the MGMT methylation status. And I also like to thank Sarah and Luis, and who is very enthusiastic supporter and sending us the cases, and I really enjoyed to talk to them about the cases. And also, a lot of help from Ken from NCI, and they have adopted this platform in their practice, and is invaluable resources for me if I have questions for interpretation. Of course, DKFZ for the classifier and the Illumina for the technical support. Thank you very much.